So now we're ready to tackle the structure of various biomolecules. And we'll talk about the four major classes of biomolecules found in life forms and look at the chemical building blocks of life. And this treatment, in this lecture, we're looking at this rather superficially in kind of a way that will just establish the nomenclature for later use when we talk about experiments and biochemistry. So we have to get the basic structures of biomolecules under our belts first. So let's go ahead and delve right into this. We are carbon-based life forms, and in chemistry, organic chemistry is the study of carbon and the way it forms molecules. So we are organic beings, and we are carbon-based life forms, therefore. And the study of life is largely the study of organic chemistry. So biological molecules are primarily of carbon, but bonded carbon forms bonds with oxygen, nitrogen, sulfur, phosphorus, or hydrogen. And these elements can also bond with each other, as we will see. Now, carbon can form up to 4 covalent bonds, and it would like to form 4 bonds whenever it can. And you can understand that very easily knowing the rules that govern atomic bonding. So if we look at carbon, here, it has 6 protons, 6 neutrons, and 6 electrons. So we can look at the configuration of those electrons easily when you understand the electron configuration. We have, at the first energy level, we have filled the s orbital with 2 electrons. At the second energy level, we have filled the s orbital with 2 electrons. And at the second energy level, there are 6 possible electrons in the various p orbitals, x, y, and z, but we only have 2. And that's 2 electrons plus 2 electrons plus 2 electrons is 6 electrons. So in its valence shell, carbon would like to acquire 4 more electrons to fill its outer shell. And then it would be 1s2, 2s2, 2p6, and the valence shell would be filled. So carbon wants to share 4 electrons with other molecules and therefore form 4 bonds. And hydrocarbons now, if we define hydrocarbons, it's a molecule that only has carbon and hydrogen in it. And there are many hydrocarbons in biology. By definition, these are nonpolar molecules. Hydrocarbons are nonpolar and therefore do not mix with water. Excuse me, we'll talk about now some functional groups and the chemical properties that the functional groups add. So when we talk about functional groups, we're adding other elements into a simple hydrocarbon, and that will dictate how those functional groups behave in a chemical sense and in biological systems. So let's get some basic functional group nomenclature down. Let's study this for a little bit here. So a hydroxyl group we will come across often. The structural formula for a hydroxyl group is just OH, and that O is bonded to something, usually a carbon, but it could be something else. And here's an example in ethanol, alcohol that we drink sometimes, some of us. Here's the OH group, and that OH group lends some polarity to ethanol. If that were just a hydrogen, this would be ethane and would be very, very nonpolar. But because of that oxygen, holding electrons more tightly than that hydrogen, that we have a polar covalent bond there. That lends some polarity to the ethanol molecule, and ethanol mixes with water. Otherwise, we wouldn't be able to drink it in wine or beer or anything like that. So we find the OH group, not ethanol, but we find the OH group in many different organic compounds in cells. Carbohydrates, very important class of molecules, proteins, nucleic acids, and lipids. In fact, these 4 types of molecules are the 4 major classes of biomolecules that we're now going to be dealing with. Carbohydrates, proteins, nucleic acids, and lipids, or fats. And we'll be talking about those shortly. Here is a functional group called the carbonyl group. And here we have carbon bonded to 2 other atoms over here, usually carbon, but it could be something else, and double bonded to oxygen. And so carbon now has 1, 2, 3, 4 bonds. Its valence shell the octet rule for carbon is satisfied. Oxygen's octet rule is satisfied because oxygen, as you know, can form 2 bonds. And so this is the carbonyl group, carbon double bonded to oxygen. And here's an example of an organic molecule, acetaldehyde, that has a carbonyl group in it. We find carbonyl groups in carbohydrates and nucleic acids, primarily in carbohydrates and nucleic acids. The carboxyl group, very important group. Here we have carbon bonded to, say, another carbon, double bonded to oxygen, and singly bonded to a hydroxyl group. Here's our hydroxyl group again. And we find this in proteins, in amino acids that make up proteins, and fats. We find the carboxyl group in lipids as well. And here's an example in acetic acid. Here is the carboxyl group, C double bond O and OH. The amino group is nitrogen bonded to 2 hydrogens. We find that, for example, in the amino acid alanine, we find the amino group in proteins and nucleic acids. Here's the amino group attached to a carbon, and this is an amino acid that we find in our proteins. And we'll talk more about that shortly. The sulfhydryl group is sulfur bonded to hydrogen, and that would be bonded over here to carbon, say. Here's another amino acid, cysteine, which we find the sulfhydryl group attached to the carbon. It looks like it's attached to the hydrogen, but actually these hydrogens are up here and here, and that sulfur is bonded to the carbon. We just write it that way for convenience sake. So don't get confused. That sulfur is actually bonded to that carbon. And this sulfhydryl group is very, very important in maintaining the structure of certain proteins. And we find it in proteins. The phosphate group, very important in nucleic acids. A phosphate group is a phosphorus atom attached to oxygens. And these oxygens are often negatively charged. They've donated hydrogens to solution. And so phosphates are, in a way, acids. If these were hydrogens attached here, these oxygens would donate their hydrogens to solution. And they actually are the reason that nucleic acids are acids, because the phosphates, the hydrogens on the oxygens of the phosphates, donate their hydrogens to solution, the definition of an acid. Here is such a phosphate on glycerol phosphate. We'll give examples of the use of glycerol when we get to fatty acids and lipid and phospholipids. Here's the methyl group. Methyl group is a pure hydrocarbon. Carbon attached to 3 H's, and let's say it was attached to another carbon over here. So in the amino acid alanine, we find that methyl group for alanine, and we find the methyl group in proteins. The methyl group is very nonpolar and therefore water-hating or hydrophobic. It doesn't mix well with water. Whereas phosphates love water because it's very polar due to these negative charges. Sulfhydryl group also has some polarity to it. The amino group has polarity to it. The carboxyl group has polarity to it. These oxygens are highly electronegative and therefore engage in non-polar covalent bonding. The carbonyl group is polar, as is the hydroxyl group. So really, out of these functional groups, only the methyl group is a true hydrocarbon and therefore is hydrophobic, water-hating. All these others are water-loving and are polar to one degree or another. So those are our major functional groups, and now we're ready to tackle whole molecules. And to start off, let's tackle a class of whole molecules called isomers. And isomers are molecules with the same molecular formula, meaning they have the same numbers of atoms and the same types of atoms. Structural isomers differ in how the carbon skeleton of the molecule, of the biomolecule, is arranged, whereas stereoisomers differ how the atoms are arranged in space with respect to each other. And a subclass of stereoisomers are called enantiomers. And enantiomers are a very special class of molecules. They are mirror images of each other, like your hands are mirror images of each other. So they are a subclass of stereoisomers. And because of that, we have a right hand and a left hand. Enantiomers are what we call chiral. That is, they have a handedness to them. We can call them left or right. And we call them left or right, L or D, D for dextro, right, L for levo, left. We call them L or D, that is left or right, based on how they act on polarized light. The L forms bend polarized planed light to the left. The dextro, the right-handed versions of enantiomers, shift the plane of polarized light to the right. So just remember that enantiomers are mirror images of each other. And examples are the L amino acids versus the D amino acids. Turns out that our proteins are composed exclusively, biological proteins are composed exclusively of L amino acids. Only the left-handed form of amino acids are found in our proteins. Apparently early in the evolution of life, the enzymes that were charged with synthesizing proteins by joining amino acids together could only join L amino acids together, and life went off on that pathway of only having L amino acids in their proteins. And in our sugars, our carbohydrate sugars are primarily of the D form, the right-handed form of the sugars, primarily. Not exclusively, but primarily. So here's an example. Excuse me. Here's the sugar glucose, and here's another sugar, fructose. And glucose and fructose are structural isomers of each other. As you can see, they have the same numbers of molecules and the same types of molecules, but the skeleton, the carbon skeleton, has been rearranged. Here we have carbon double bonded to oxygen, whereas over here in glucose, carbon is bonded to a hydroxyl group. So here we have a carbonyl group, and here we have a hydroxyl group. And the carbonyl group is here. Whereas glucose is an isomer of galactose, but they are stereoisomers. And you can see that they are, in this case, they are the arrangement in space of the hydroxyl group is different in galactose than it is in glucose. Now, these are not mirror images of each other. They're stereoisomers, but they are not enantiomers. We'll come across enantiomers now. Here's examples of enantiomers. So this molecule, we have a carbon attached to 4 other atoms here, or these could be groups of atoms, it doesn't matter. These are the same groups of atoms attached to this carbon. But they're arranged in space such that this molecule is a mirror image of that molecule in the same way that your hands are mirror images of each other. And that is very important when we talk about the proteins, the L amino acids in proteins. Now, macromolecules, moving on, macromolecules are polymers, meaning that they are built by linking monomers together. So much like you would take Lego building blocks and join them together and build a long chain of Lego building blocks, that is what a polymer is. When we polymerize building blocks together to form a long chain, we are building a polymer from the individual building blocks, which we call monomers. So monomer then is a small, similar chemical subunit that can be joined together to form a polymer, which constitute the large molecules found in cells, the macromolecules. So, and in our next part of this lecture, we'll tackle the carbohydrates, nucleic acids, proteins, and lipids, and we'll give examples of these. And we will talk about a particular type of chemical reaction that is used in the polymerization of monomers into polymers. And that's what we'll pick up with in the next part.